And so today, welcome to this week's Fragile Nucleosome. It's my pleasure to introduce Rachel Hoffman, who's currently a postdoctoral science at Nationwide Children's Hospital. She got her start in research way back as an undergrad, it looks like, here close to me at UNC Chapel Hill, and then did her PhD in the McAlpine lab at Duke University. And after that, she moved to Nationwide Children's Hospital to work in the Stanton lab, where today she's going to tell us about some real cool work she's doing, looking at fusion transcription factor in rhabdomyosarcoma. So, Rachel, please take it away.
Great, thank you. Oh, there we go. Okay, so today I'm going to tell you about my work on a transcription factor in a pediatric cancer. So today I just want to start out with some basic context before I really dive into the details of the cancer I study. Just broadly, what are the differences between pediatric and adult tumors? One of the major differences is that pediatric cancers tend to have a lower mutational burden than adult cancers. As demonstrated by this plot of coding mutations per megabase across a range of pediatric and adult cancers. And given the lower number of actual genetic changes in pediatric tumors, epigenetic state is really important to understand in these diseases because just when we're trying to understand mechanism and look towards developing new therapeutics, which are sorely needed in many of these pediatric cancers. And many pediatric cancers have genetic mutations that actually alter chromatin in some way, which is the case for the disease that I study. So I study fusion-positive rhabdomyosarcoma, which is driven by a translocation that produces a fusion between PAX3 and FOXO1, and it encodes an oncogenic fusion transcription factor. And just to give you an idea of what this factor is like, so we have the DNA binding domain of PAX3 fully preserved in the fusion, and we don't have the DNA binding domain of FOXO1 fully preserved in the fusion. So what's previously been found is that PAX3 FOXO1 frequently binds to a composite motif consisting of the paired and homeobox motifs corresponding to PAX3's paired and homeobox domains. It's really important to understand this fusion transcription factor and what it does on chromatin because it's a major driver in the disease and likely the first, the translocation that encodes it is likely the first mutation that occurs in tumor evolution. So prior work in our lab has found that PAX3 FOXO1 has what we call pioneer function, meaning that it doesn't just bind to open, accessible regions of the genome, but it can also access closed heterochromatic regions. And in some cases, we found that it can actually induce accessibility at those more closed regions of the genome. As you might imagine, this could be really useful for altering the epigenome and driving cells towards pediatric cancer cell state. And so we also know PAX3 FOXO1 can modulate the oncogenic character of cells. And prior studies actually found that the dosage or expression level of PAX3 FOXO1 is really important for the precise kind of oncogenic characteristics of cells in which it's expressed. So in one model, it was found that PAX3 FOXO1 has more of a tumor propagating or migratory effect. It gives more migratory character to cells in which it's expressed at a low level. And high PAX3 FOXO1 expression is actually more associated with tumor initiation. So we know that broadly PAX3 FOXO1 expression level seems to modulate oncogenic character, but we don't actually understand how on a chromatin level it can modify cell state. So that's one of the major questions I've been trying to investigate is how does PAX3 FOXO1 actually modulate cell state in rhabdomyosarcoma to give us this range of effects across PAX3 FOXO1 expression level. So we of course hypothesized that differences in chromatin recognition with PAX3 FOXO1 expression level alter gene regulation and then the resulting cell state. So I've been able to study this in a human myoblast system that was developed by our collaborators at the NCI and Dr. Fred Barr's lab. And The system has been really useful because it has a doxycycline-inducible PAX3-FOXO1. And just to demonstrate kind of the utility of this system, when we add different levels of PAX3-FOXO1, or sorry, doxycycline to cell media, we get a range of PAX3-FOXO1 protein expression. And this is the case regardless of whether we induce PAX3-FOXO1 induction for 8, 24 hours, or even up to 2 weeks, we get this range of expression. So this has been a really useful model for us. And I do want to note that the context of the human myoblast is also like a really great cell context to examine. It's one of the potential cells of origin for rhabdomyosarcoma, and so allows us to really model fusion rhabdomyosarcoma. And I'll talk about that a little bit more later. So initially, to determine whether PAX3-FOXO1 in this model can actually modulate oncogenic character of cells, we did a focus formation assay across a range of PAX3-FOXO1 expression levels, and we find with increasing PAX3-FOXO1 dosage that we get maximal focus formation, which reflects like the oncogenic character of the cells at an intermediate dosage of PAX3-FOXO1, and then if we add more protein expression, we actually get decreasing focus formation. So there's kind of a point at which in this system, we don't get additional focus formation. And so there's kind of this maximum intermediate point. I'm emphasizing this because this trend will come back a few times during the talk. And so I want you to think back to this in a few slides. But of course, we really wanted to know, like, is there any heterogeneity in this system? When we see PAX3-FOXO1 modulating oncogenic potential, is that actually altering cell state in a way that's more heterogeneous, or is that just kind of like one note? So we looked at single cell RNA sequencing, or we did single cell RNA sequencing in the system at different expression conditions. So we did different time points and then also different dosages of PAX3-FOXO1. And as you can probably appreciate from these UMAP plots, at a shorter induction time point, we really see less heterogeneity of expression, of gene expression. Only 2 major populations, which really just reflect like cycling cells, like you'd expect in like a cell culture model. But after 2 weeks of expression of PAX3-FOXO1, we actually do see more heterogeneity. We see multiple cell populations based on gene expression, and some of these, importantly, actually correspond to actual populations in fusion-positive tumors that have been identified through single-cell sequencing. So this really surprisingly validated our model, I think, in more than we expected, to be honest. We also did this 2-week induction across multiple PAX3-FOXO1 expression levels. And so we also found that these cell populations we identified occur in different proportions based on PAX3-FOXO1 dosage. So some of them, like this progenitor-like population, are more prominent at the no induction condition. We find this differentiated or myogenic cluster maximally present in the population at low expression of PAX3-FOXO1, and then we actually find the aptly named P3F specific cluster, which is characterized by high expression of PAX3-FOXO1 target genes at high dosage most frequently. So PAX3-FOXO1 does seem to be able to modulate cell state or cell identity, at least on this through the single cell sequencing. We can observe that, but what's actually going on the chromatin level to drive what we've observed, both in our single cell data and the focus formation data I showed. So we did a ChIP-seq for PAX3-FOXO1 across a range of dosages, and this is just to show you what a little snapshot of what this data looks like. This is at the FGFR4 locus, which is a key site or a key target of PAX3-FOXO1, and we see 2 PAX3-FOXO1 peaks here, which are present across all dosages. So we don't see any major changes in like peak width or peak shape. There are many sites that are commonly occupied by PAX3-FOXO1 regardless of dosage. However, we do find when we look at all of the genomic binding sites for PAX3-FOXO1 that we identified across replicates, across different PAX3-FOXO1 dosages, that we have a different number of genomic binding sites based on dosage. So as you can see, at a low expression level of PAX3-FOXO1, we have fewer binding sites that then increases at this intermediate dosage, and then that decreases actually with increasing dosage. So this is reminiscent of that kind of increasing to decreasing trend that I mentioned with the focus formation assay, which is really interesting. That kind of lines up on a chromatin level. And we, of course, are really curious how paxifoxal-1 might be binding, like how we actually have modulation of the binding sites that are used by paxifoxal-1 based on dosage. And so we looked at motifs that might distinguish these different conditions. And indeed, we did find some really intriguing motif differences enriched at these, excuse me, at these different dosages. So we found BZIP motifs highly enriched at the lower expression of paxifoxal-1. So those, for some reason, we see motifs typically bound by AP1 complex members enriched at paxifoxal-1 sites, specifically at low dosage. We hypothesized that this could either, of course, reflect cooperativity with the BZIP factor, or we actually also hypothesized that given there's some similarity between BZIP motifs and the paxifoxal-1 composite motif, that paxifoxal-1 itself could be doing some kind of partial motif recognition at these BZIP motifs at low dosage. At intermediate to high expression level of paxifoxal-1, we find an enrichment of basic helix loop helix motifs, and we do think these correspond to myogenic basic helix loop helix factors that we already know are really important in fusion positive rhabdomyosarcoma. So these would be factors like MyoD and MyoG. So we think those might be more often present near paxifoxal-1 at its binding sites at intermediate to high dosage of paxifoxal-1. Then finally, at high expression of paxifoxal-1, we find its composite motif more enriched. So paxifoxal-1 seems to recognize this composite motif more frequently at this higher dosage. And this is certainly a more complex motif. So maybe there's some difference in binding mode for paxifoxal-1 based on dosage. There is potentially also differences in cooperativity based on dosage, which is really intriguing. And we're definitely following that up biochemically. But we, at this point, also really wanted to know what's the functional output of this modulation of paxifoxal-1 chromatin recognition. And so I, to answer this question, turned to a RNA-seq data set that we have that is matched to this ChIP-seq data set. And we looked specifically at genes that are near paxifoxal-1 binding sites to try to just narrow our search to like genes that are actually most likely directly regulated by paxifoxal-1. And because paxifoxal-1 is an activating factor, is known to be in the literature, we expected paxifoxal-1 dosage to mainly increase transcription at genes that have nearby paxifoxal-1 ChIP-seq sites, but that actually isn't what we observe. As you can see, based on k-means clustering, we observe these different trends for paxifoxal-1. Paxifoxal-1's like its role in respect to regulating dosage, regulating transcription based on dosage. And just to highlight a few of these, we actually see that some genes decrease expression with increasing paxifoxal-1 dosage. And some of the gene sets that come up amongst these genes are epithelial mesenchymal transition, hypoxia, TNF alpha signaling, which is really intriguing given we're interested in how paxifoxal-1 can modulate the kind of the oncogenic character of cells based on dosage. And also we find these intermediate trends where paxifoxal-1 dosage, in the case of this fifth cluster here, it upregulates these genes to an intermediate. Well, I should say, they are most highly expressed in an intermediate dosage. And those tend to be myogenic genes, and that's really intriguing given the myogenic basic helix loop helix motifs that we observed at intermediate dosage. Also corresponds to kind of that like intermediate maximal trend that we've been observing for some of the other data sets. And then finally, we do see some genes that increase in expression with paxifoxal-1 dosage like we kind of expected initially. Many of these genes are actually cell cycle related genes. So some gene sets we get enriched are E2F targets, which are S-phase genes, G2M checkpoint or mitotic spindle. Those would be M-phase related genes. And I was really interested to follow up whether cell cycle progression is really modulated by paxifoxone dosage, or whether this is just like transcriptional, because the transcription doesn't really show that there's modulation of these processes, of course. So I did look at the proportion of S-phase cells in the population with increasing paxifoxone dosage. So we do find an increasing proportion of S-phase cells in the population based on dosage. So one thing I haven't mentioned to this point is that paxifoxone actually does not increase cell proliferation with increasing dosage. It actually decreases it. So we think this increase in S-phase cells doesn't represent like additional cycling, faster cycling of cells. We hypothesize that it could represent a more prolonged S-phase, which would be very interesting. Paxifoxone transcriptionally regulating these S-phase genes and then with an outcome of prolonging S-phase progression. However, this also really got us thinking about paxifoxone's impact on S-phase more broadly. And given our interest in paxifoxone's pioneer function, ability to modulate cell state, we have also turned to investigating whether paxifoxone could actually modulate the process of chromatin maturation. And just as kind of a brief review, ahead of a replication fork, chromatin has to be disassembled, and then it's reassembled behind the fork. And during the process of chromatin maturation, histone modification levels, accessibility patterns, et cetera, have to be restored over time during maturation. So we wondered if paxifoxone, a pioneer factor, could actually perturb that process of maturation. And so I've initially investigated this with a technique called RepliAttack-seq, developed by Anya Groth's group. And this has allowed us to examine specific time points during chromatin maturation to assess how chromatin accessibility recovers after replication. So we're using a nascent chromatin time point, 10 minutes after replication fork passage, and then a mature or maturing chromatin time point, 2 hours after fork passage. So 2 distinct time points where we can really see how accessibility recovers and if it's actually modulated by paxifoxone. And so what we found when we looked at nascent chromatin accessibility across the genome is that with increasing paxifoxone expression, we actually see an increase in the number of accessible sites across replicates, which was really intriguing. And of course, in mature chromatin, there are more accessible sites than in nascent chromatin, like has been shown in the literature. And we also don't see that dosage trend in mature chromatin, likely because chromatin has had more time to mature. There are many factors that contribute to chromatin accessibility, but we really see that dosage effect in nascent chromatin. So we wanted to see if there are any signs that we could surmise of paxifoxone's influence on these accessible sites. And so we did motif analysis, and we found basic helix loop helix motifs enriched at only the conditions where paxifoxone is expressed in both nascent and mature chromatin. Given the basic helix loop helix motifs we have found in our data set at paxifoxone ChIP-seq sites and the importance of myogenic factors that I've talked about already, we do think this is a sign that paxifoxone might be modulating accessibility at these sites, though it's not like directly showing the composite motif or anything like that. We further looked at all the accessible sites and were able to categorize them into either sites that are either dependent on paxifoxone for their accessibility restoration or independent of paxifoxone. And I'm going back to this site at FGFR4 to look at these paxifoxone sites I mentioned earlier, because they actually are really representative of this trend. So you can see at this first site here, we have accessibility in nascent mature chromatin, even in the absence of paxifoxone induction. So this site is actually independent of paxifoxone for chromatin accessibility restoration, even though paxifoxone does bind that site at some point. And we also find, interestingly, in contrast at this site here, that paxifoxal-1 is necessary for chromatin accessibility to be restored. We only see accessibility when paxifoxal-1 is expressed in the system. And so we can categorize all accessible sites based on those two categories. And I want to end by zooming out on all of the paxifoxal-1 ChIP-seq sites we identified, and look at chromatin accessibility after replication at these sites. Because if we're looking for paxifoxal-1's influence on chromatin maturation, maybe we'd find it right at these sites. And so as you can see, in nascent chromatin, there's just different baseline accessibility across these sites. And so we've clustered based on kind of that initial accessibility. But regardless of the initial accessibility of these sites, without paxifoxal induction, we do see an increase in accessibility at these sites with paxifoxal expression, especially high levels of paxifoxal expression. And we see this in mature chromatin as well. I'm most intrigued, honestly, by these sites that are really inaccessible in the absence of paxifoxal induction, and then we see increasing accessibility at high paxifoxal expression. Really excited to follow up on what's actually going on at those sites, what this modulation of accessibility rapidly after application, what the consequences of that might be for cell state. And I do want to note that I've shown you data that suggests paxifoxal seems to potentially modulate chromatin maturation. I haven't been able to demonstrate yet whether that is directly by paxifoxal finding the genome at these sites or through paxifoxal transcriptionally regulating other factors that bind to these sites. And so I think my next step is definitely to do a more factor-specific experiment to look at whether paxifoxal itself really binds nascent chromatin, and to really explore like what the consequences could be on cell state when it modulates this process. So just to sum up what I've told you today, we have been able to demonstrate in our model system that paxifoxal expression level modulates the oncogenic character of cells as measured through focus formation, and that this also is accompanied by modulation of gene expression, different interesting gene sets relevant in rhabdomyosarcoma are modulated by paxifoxal expression, both in the bulk cell RNA-seq I showed, and we have different populations in our single cell data that are also modulated in proportion by paxifoxal dosage. Underlying that, we see motif recognition differences, potentially, or at least different cooperativity with other factors at paxifoxal sites based on dosage. We see the most paxifoxal sites bound in the genome at intermediate dosage of paxifoxal, intriguingly, and paxifoxal might be able to modulate cell state in rhabdomyosarcoma by interfering with chromatin maturation, especially at high expression level of paxifoxal. So with that, I want to acknowledge everyone who supported me in the Stanton Lab, including especially Ben for his mentorship over the past few years. I really want to acknowledge our collaborators at NCI for supporting this work, especially Dr. Fred Barr. There's many people I could recognize at Ohio State, Nationwide Children's, but I'll just mention Tim, Genevieve, and Christine for serving on my committee, and acknowledge my funding, and happy to take any questions.
Right. That was great. Thanks so much. Again, if you have any questions, please raise your hand and we can call on you. We've already got several questions in the Q&A box, so I'll start with one of those. The first is, how do the gene expression changes relate to the binding of PAX3 and FOXO? Since there are more binding sites at intermediate dosages, are there more expression changes under those conditions?
So yeah, based on the data I showed, kind of a lot of gene sets are reaching like an intermediate expression level relative to dosage under that condition. So I guess I don't know if I have the information to tell if like gene expression is like increased on like a holistic level overall, but we do see kind of like a lot of kind of intermediate expression. So I would say that it's possible.
Then we have a question from Genevieve Farrell, who says, well, I guess she finishes by saying, thank you, wonderful work. And then says, did you try to express PAX3 FOXO1 and G1 arrested cells? Or did you check to see whether slowing down in S might be due to DNA damage?
Yeah, so yeah, it's a really great question. We have tried to look for like markers of replication stress and DNA damage, and I didn't find any preliminarily. It's still something, yeah, like there wasn't anything really striking there, so it doesn't seem to be exactly what's going on. We haven't tried this in G1 arrested cells, yeah.
And let's see, we're running pretty close on time, so I will leave the rest of the questions in the Q&A for you to answer after this. If you have further questions for Rachel, please go ahead and keep typing in the Q&A, and she can answer those as she gets going. But now I'll pass it over to Skye to introduce our next speaker.